Lecture number 8. On this lecture we're going to cover the structure of our immune system. So our immune system consists of 3 lines of defense. On the previous lecture we already talked about the first line. It's called non-specific surface defense, and it consists of 3 parts. Can be structural, mechanical, and biochemical. So example of structural part will be structure or anatomy of the skin or mucous membranes. Example of mechanical part of the first line will be, for example, urine. When it goes through the urinary tract, it removes microorganisms from the surfaces of the mucous membranes. Another example of mechanical part of the first line will be mucociliary epithelial. It lines upper respiratory tract. Peristalsis or bowel movements, another example of mechanical part of the first line. And finally, biochemical part of the first line. An example of biochemical part will be stomach acid, bile salts, and multiple enzymes that can be found in saliva, perspiration, or tears. The second line of our immune system is called non-specific innate immunity. So as you see, the second line is non-specific line as well. And we're going to talk about 5 main parts of innate immunity, and those are phagocytosis, inflammation, the complement system, interference, and natural killer or NK cells. And finally, the last line is specific adaptive immunity. As you see, this is the only specific line in our immune system, and it's presented by T cells, B cells, and antibodies that are produced by B cells. On this slide, once again, in this chart, colorful chart, you see the main characteristics of 3 lines of defense. And as you see, the first 2 lines are non-specific lines. It's non-specific surface defense and innate immunity. And the third line is the only specific line in our immune system, and it's presented by T cells, and antibodies. This chart compares for you innate immunity, the second line, to adaptive immunity, or the third line of defense. So let's start with the speed of response. Innate immunity is fast, it's rapid. So basically it starts working within few, sometimes even minutes. Adaptive immunity, slow. It takes a while, it takes sometimes couple weeks to make antibodies for adaptive immune system. Distribution in nature. Innate immunity present in all animals. Adaptive immunity present only in vertebrates. Variation over the time. Innate immunity never changes. It stays the same. That means you're born with certain type of immunity, you'll die with the same type of immunity. On the other hand, adaptive immunity changes all the time. Every time when we get exposed to the new antigen, our adaptive immune system will make new antibodies to those new specific antigens. The next characteristic, means of recognizing pathogens. Innate immunity recognizes only common molecules on the pathogens. For example, peptidoglycan on the bacterial cell walls, or LPS on the surface of the outer membrane of gram-negative bacteria, or chitin in fungi. Adaptive immunity recognizes unique or specific molecules. It means it can recognize each strain of certain bacterial species. numbers of molecules recognized, only few common molecules for the innate immunity, and the number of antigens that our adaptive immunity recognizes during our life cannot be even calculated. And finally, change during the response. Innate immunity always stays the same. It's constant, never changes. On the other hand, adaptive immunity improves during response. And here we're going to talk about primary immune response and the secondary immune response, and you will see how adaptive immunity improves. Both the innate immunity and adaptive immunity depend upon the actions of the leukocytes or white blood cells. There are more than 10 different kinds of leukocytes we know, and all of them can be divided into two main groups. A, granulocytic, means have no granules in cytoplasm, and granulocytic, it means they have granules in their cytoplasm. Let's talk about agranulocytes. Those are lymphocytes, the smallest white blood cells, and monocytes. And by the way, we call them monocytes when they are in the bloodstream, but when they leave blood and go to the tissue, they become macrophages. Therefore, we can say that monocytes give rise to macrophages. And then the second group, those are granulocytes. Granulocytes have granules in the cytoplasm. So those are neutrophils, eosinophils, and basophils. On this slide, you see how white blood cells look like under microscope after we dye them, after we give them some color. So as you see, basophils are purple white blood cells. Neutrophils, kind of pinkish purple. Eosinophils are orangey purple. Monocytes are large cells. And you see, this is what we call macrophage because it's already engulfed some pathogens. And then finally, the last one is a lymphocyte. Lymphocytes are the smallest white blood cells. The most common blood test that actually we perform for each patient is CBC with diff, which means complete blood count with differential. On this slide, you see differential part of that blood test. You see percentage of each type of the white blood cell in the blood. So neutrophils. Neutrophils are the most numerous white blood cells. You see we have 60 to 70% of this type, and they protect us against bacterial infections. And then the next one, those are lymphocytes, from 20 to 25%. They protect us against viral infections. And then the next group, those are monocytes, from 3 to 8%. Remember, monocytes give rise to macrophages. And then eosinophils, from 2 to 4%. Eosinophils participate in allergic reactions and also protect us against the helminthic infection. And finally, the last group, those are basophils, up to 1%. We don't know much about the function of basophils, but we know that they participate in allergic reactions with eosinophils, and also they are part of inflammatory reactions.